Crossing over in meiosis 1 is required for homologous chromosomes to properly align during metaphase and segregate during the first cell division. So the question starts by telling you the importance of crossing over, meaning that we're going to be going over crossing over probably during the rest of the question. AI, describe the function of the S phase of interphase. This is where DNA gets replicated. Okay. Some regions of a chromosome called hotspots display a higher frequency of crossing over than other regions do. Crossing over is suppressed, which means does not happen in the chromosomal regions near the centromere. The centromere region of a duplicated chromosome includes a collection of proteins that form a structure called the kinetochore. Scientists hypothesize that one or more of those kinetochore proteins are responsible for suppressing crossing over around the centromere. So now we know that one or more of those kinetochore proteins are responsible for the suppression of the crossing over near the centromere region, which is the black dot there. To investigate their hypothesis, scientists modified chromosome 8 in yeast so that in each cell, one chromosome from the pair of the homologous chromosome contained the gene giving you red color. The other chromosome contained the gene giving you green color. So here we have red, green, and green. But this is before crossing over. However, after crossing over, what we see is that the edges remain the same. So red remains red, green remains green, but one of the chromatids on the middle loses, so no color. Other one will now have two colors, red plus green. Double I, explain why some haploid cells formed after meiosis in this experiment will have only one fluorescent marker. Why would that be? Okay, so to have one fluorescent marker, you would either have to be red or you would have to be green, which basically means you were red originally or green originally and nothing has happened. That means what? They did not exchange any gene segments. So crossing over happened, but without them being included. Okay, so basically the chromatid with a single color fluorescent marker is the one that didn't undergo crossing over. So the haploid cell ended up receiving this specific chromatid. The scientists then investigated whether attaching individual kinetochore proteins to a specific DNA sequence present in a non-crossing over hotspot would affect the frequency of the crossover at this specific location. In their first experiment, they examined 3 groups of yeast cells containing the modified chromosome 8. Group 1 contained no kinetochore proteins, which is this one. This would obviously serve as our control because it does not have any ability of having kinetochore. That means that the crossing over is not going to be affected. The second group contained the kinetochore protein CTF, and the third group contained the kinetochore protein IML. For each group, the scientists determined the frequency of crossing over between the red and the green. To determine the frequency, the scientists added the number of the cells emitting both red and green, which is basically the ones that only one chromatid here is able to emit both red and green. Okay. And divided that by the total number of cells. Okay. BI identified the control group for the scientist's first experiment shown in figure 2. That would be the group that is called none. Right? It's called none. BII, in a follow-up experiment, the scientists created a modified version of CTF. Okay, which is this one. And CTF, if you recall, was actually a kinetochore protein that was suspected of suppressing crossover. The scientists created a modified version of CTF where its DNA binding region has been removed. That means that it is unable to bind to the DNA. They compared the frequency of the crossover in these cells in the presence and the absence. So, because you're able to control whether it's present or absent, this is called the independent variable. Okay. And also, they would control the presence or the absence of the modified CTF, which would also be our second independent variable. In a follow-up experiment, justify why the scientists used a modified CTF protein that is unable to bind the DNA as a control. So, of course, they remove the DNA binding portion to identify its function. The best way in biology to know the function of something is to simply remove it. For example, someone's going to say, why would a car have 4 tires? I think 2 tires will work. So the scientist would say, okay, remove 2 tires and see what happens. Okay, so the same thing here. So this would allow the scientists to assess the importance of the DNA binding portion of CTF on its ability to affect crossover frequency. Okay, triple I. Identify the independent variable in the follow-up experiment. Like I said, you can either choose the presence and the absence of the unmodified CTF, or you could choose to say the presence and the absence of the modified CTF protein. It's obviously up to you which one to choose. They're both going to be equally correct. C. Based on figure 2, describe the effect on the frequency of crossing over when CTF is attached to chromosome 8 hotspot compared to the effect when IML is attached. So, of course, to know the effect of something, you must obviously compare it to the control. So compared to the control, we can tell that the CTF frequency of crossing over has no overlap with the error bars of that of none. And it's going to be significantly less. So we're going to say that CTF has statistically decreased crossing over frequency compared to group none, while, as you can tell, IML and none are overlapping. While IML and none do not differ in their crossover frequency, so IML had no effect. Okay, DI, predict the effect on the number of copies of chromosome 8 likely to be present in the resulting daughter cell when CTF is attached. Well, we know that CTF was able to decrease the frequency of crossing over. Okay. It decreased the frequency of crossing over. And if you recall, in the beginning of the question, we were told very explicitly that crossing over is responsible for the proper alignment and segregation of homologous chromosomes. Hint that if crossing over is reduced, you're going to have less proper segregation of homologous chromosomes, a phenomenon called non-disjunction. So how do we go about doing this? So let me just ask you a question. And of course, you always answer by repeating the question, but don't do that this time. If we have less crossing over, would that be a good thing or a bad thing?
Bad thing.
Bad thing, okay. So what do you expect if you have non-disjunction in meiosis 1? So you know how the number of the gametes has to be 1?
Yeah.
N. Will it be different now if there is non-disjunction?
Yes.
What is it going to be?
I think it's gonna be more.
Either more or less
yeah.
by 1. So that means we're going to say now the daughter cells will have 1 more chromosome or 1 less chromosome than normal number of chromosomes. That's for the predict part. Of course, now they're going to say justify that. So now we're going to justify it by saying CTF decreases crossover frequency. So this can lead to non-disjunction in meiosis 1. III, explain how the presence of hotspots in figure 1 could increase the likelihood that a population will survive in the presence of selective pressures. We were told in the first page that hotspots increase the amount of crossing over, and as we know from our background knowledge, more crossing over would lead to more genetic diversity, which is a good thing. So we're going to say that hotspots increase the frequency of crossover. So this leads to more genetic diversity, so the population can benefit from natural selection.
I would have just wrote more genetic diversity.
Well, they should explain. Will benefit or experience natural selection, basically, because natural selection works on variation. Variation is like the raw material. If it doesn't exist, selection won't work. Okay, so here we're talking about to investigate how the increase in environmental temperatures affect the metabolism of certain organisms. Researchers incubated liver cells from toads, which is like a frog, at different temperatures. So here right away, we can tell that different temperatures is going to be our independent variable. And they measured two markers of metabolic activity. So we can tell right away that what they're measured is going to be your dependent variable. A. Describe the role of water in the hydrolysis of ATP. This is just the background knowledge information.
Which is, breaking down the polymers, basically, or breaking down the ATP.
Yeah, is used to break down ATP. Good. So here we have our table. Whenever you see this plus and minus, it means that we have error bars. Are you good with the graphing now?
I'm kind of good.
Okay.
But Mr. Sometimes I'm confused with line graphs and bar graphs. Like, I don't know which one to...
Well, look, people always ask me this question, so it's not just you. A line graph is when you have time in 5 or more, different times.
Okay.
So if you have 5 or more different times, we're talking about line graph. Or if you have 5 or more different concentrations, of course, as your independent variable. So if you have time as the independent variable or concentration with 5 or more data points, that's your line graph. Otherwise, it's going to be a bar graph. So do we have here time as the x-axis? No. Is it concentration on the x-axis? No.
No.
So we're going to actually have to build here 6 bars. It's a bar graph of 6 bars. Okay. So now, using the template and the space provided, construct a bar graph. Actually, here they tell you it's a bar graph. But we should take care of the plus and the minus, right? That's the most important thing. So now, do me a favor and use your calculator and help me with the plus and the minus. I'll do some and you do some, okay? So like here, you should do 10.6, 15. Okay, so I'm minusing and plussing, right?
10.6 minus 15.
No. So like basically, if you were to do 12.8 minus 2.2, you would get 10.6.
Yeah.
And if you were to do 12.8 plus 2.2, you would get 15 or 14?
12.8 plus 2.2 should be 15.
Oh, okay. So that means I will range from 10.6 to 15. So now we should do the same, the plus and minus for all of them.
So 12.6 plus 1.6, just 14.2.
And if you minus, you get 11. Okay. Here, if you plus 18.5, if you minus 14.5. If you plus 18.8, and if you minus 14.8. If you plus 22.8, and if you minus 21.3. Here, if you plus 21.87, and if you minus, how much would that be?
21.07 minus 0.8, which is 20.27.
Okay, so now I should take a picture of this data, right? And I should just plug it in my graph, bar graph. But there's no template. Weird.
I did like this for...
Let me pull up a template from my Google Drive. Okay, so here, let me, on the y-axis, right. So look, on the y-axis we have rate. Rate of consumption in the case of oxygen, and rate of synthesis in case of the ATP. Now, is the question asking for... Yeah, the question is asking for both. Exactly. Now, they both have the same unit though, right? So they both have n mole per minute per mg. N mole per minute per mg. So we're just going to say the rate. Okay. The rate and and they both have mitochondrial protein, right? So the rate of mitochondrial protein, which is going to be n mole minute mg. Now, for the x-axis, I'll be putting the temperature, right? So I'll be putting the temperature as the x-axis. Okay, now I'm going to be actually pulling up my data and trying to use it. It's going to be hard to see a bit, but we have to see it, right? Okay, so for example, I have three temperatures. I have 20 and 25 and 30, right? So I'm going to here have 20 Celsius, 25 Celsius, and 30 Celsius. Now, I'm going to have here 12.8. Now look, my least data on the y-axis have to go from 10.6 to the highest data has to be 22.8, correct? So now I should label my axes accordingly. And if I, look, do I have to start from 1?
No.
No. So I will do a double tick, we call that, and I'm going to start very close to 10. Let's just say 9. Okay? 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22. And you should, and 23. You should always cover 2/3 of your graph, meaning you should be ultimately covering, 2/3 like this, right? Or maybe a bit more. So you should always make sure to cover 2/3, because what some people do is that they graph everything in here. And of course, that's a minus. Or they got everything in here. But how about the other 2/3? So you should always make sure that you're covering 2/3 to be safe on the x and also the y-axis. Okay, now. So now here I have 12.8. So 12, 13. So 12.8 is going to be somewhere here, right? So I'm going to be drawing a bar at 12.8. I'm going to be drawing it all the way to the x-axis. And I should range from 10.6 to 15. So this is 11. So 10.6 is going to be here. And 15 is going to be here. So now I should connect them by one straight line. Cool. I took care of who? Of the 20 Celsius for the oxygen consumption. Now I'm going to have to take care of the 20 Celsius for the ATP synthesis, which starts at 12.6. The same, right? This was 12.8. This is 12.6, almost the same. So I'm going to do it a bit lower, maybe here. Draw it to the x-axis. And now I'm going to have to range from 11 to 14.2. So 11 to 14.2. Connect them by one straight line. So far, so good. But now how would I let my test grader know which one I'm referring to?
You can put dashed lines for the ATP synthesis.
Exactly. So I'm going to do what we call the color coding. I'll put a dashed line for the ATP synthesis. Later I'm going to have to do a color code once I finish this, which I will. Now we took care of the 20 Celsius. Time to go to the 25 Celsius. Let's take care of the oxygen consumption, which starts at 16.5. So 16.5, that's 16, that's 17. That's going to be somewhere here, connected to the x-axis. And we have to range from 14.5 to 18.5. So 14.5 is going to be somewhere here. And this is 18 and this is 19. 18.5 is somewhere here. Connect them by one straight line. Cool.
Yep.
And now I'm going to do the other one, which is 16.8. Again, very close to it. So here probably. And now I'm going to have to go from 14.8 to 18.8. So 14.8 to 18.8. Good. I took care of that one, also shaded. Now I have to go now to the 30 Celsius, which starts at 22.1. So 22.1, that's 20, that's 21, 22, 23. So 22.1. Let's move this guy here a bit. 22, or here maybe. Okay, you have to do this very fast in the exam, obviously. 22.1, so here would be perfect. And we have to range from 21.3 to 22.8. So from here to here. I think I made it. No, sorry. 22.8 is here. Okay. And now for the other one, we're going to have to do from 21.07, which is here. And we have to go from 20.27 to 21.87. And again, shade it. And now for the final part is the color coding. So I'm going to here tell them that if I have, a white square, then I am talking about oxygen consumption. But if I have a dark square, then I'm talking about
ATP synthesis.
Yeah, ATP synthesis. And I would like to just pull that a bit up, or not, just leave it like this. Perfect. So now they can see that. And that's what the graph looks like. Yep. That's pretty good. It's pretty good. Now they always ask a question about the graph. So now let's see what they're asking. In part B, they're asking to construct, which we did. Then they're asking to, based on the data provided, determine the temperature in Celsius at which the rate of the oxygen consumption is different from that at 25 Celsius. So look, the 25 Celsius is right here. So now we should find the rate of oxygen consumption. Oh, sorry. So it's the white one. So which temperature will be different than this one? The 20 Celsius or the 30 Celsius for the oxygen consumption?
Just the 30 Celsius.
Right, because if you have the 20 Celsius,
it's overlapping.
It's overlapping with that one, right? So it's not going to be the 20, it's going to be the 30 Celsius because there is no overlap. So yeah. So now for this one, we're going to say the 30 Celsius due to no overlap of error bars. CI, based on data in table 1, describe the effect of temperature on the rate of ATP synthesis. Okay, so back to my data. What's the effect of temperature on the rate of ATP synthesis?
Clearly has an effect as it goes up.
Right, so there's more temperature, there is more rate of ATP synthesis, correct? Because we are as low as here, we're higher, right? And there's no overlap, and then we're even higher. So more temperature means more ATP synthesis. So we're going to say more temperature leads to more ATP synthesis. Okay, very good. Based on data in table 1, calculate the average amount of oxygen consumed in N mole for 10 milligrams after 10 minutes. So look, at 25 Celsius. So now we should go back to the table and look for the 25 Celsius, right, for the oxygen consumption, which is here. Don't look at the error bars for this math, okay?
Okay.
Now look at the unit. It's nmol per minute per milligram. So right now we're going to be writing 16.5 nmol per minute per milligram. Now I think you know from math that these guys have to now become division, and they have to be flipped. So milligram has to go on top and minute has to go on the bottom. Like this. Does that make sense to you? No? Okay. So like in math we have this rule that says if you have A over B over C, it's like having A times B over C gets flipped to C over B.
Okay.
So now the same thing I'll do here. I'll be flipping the minute per milligram, make it division, and then flipping it to milligram over minute. Now here we have for 10 milligram in 10 minutes. Actually, 1 second. Let's just see what happens if I don't flip. So nmol per minute per milligram. Let's just keep it like that. For 10 milligrams, so times 10 mg, right?
Mhm.
After 10 minutes, so that times 10 minutes. Okay. So, yeah. So now what happens? The minutes and the minutes cancel, right?
Mhm.
The milligrams and the milligrams cancel. And what do you get instead? You get 1,650 nmol. Is that good?
Yeah.
Yeah. Okay, we got it. DI. Oligomycin is a compound that can block the channel protein function of ATP synthase. Predict the effect of using oligomycin on the proton gradient across the inner mitochondrial membrane. So let's just do a small recap here of what's happening with the last two steps of ATP making in the mitochondria, or last two steps of aerobic respiration. Here we have what we call the outer mitochondrial membrane, and here I have ATP synthase located on the inner mitochondrial membrane. Okay, so that's ATP synthase, and by natural movement of H+, it's going to move from the mitochondrial matrix to this space between the two membranes, which is called the intermembrane space. H+ is moving there. And then you're going to have a lot of H+ here. And now this H+ now wants to do what? Wants to move back. Through who? Through the ATP synthase. But now, if the ATP synthase is not working, can the H+ move back?
No.
No. So what's going to happen? The H+ is now going to accumulate in high amounts in this space between the two membranes. And the H+ is called the proton gradient. So if you have a lot of H+, you can have a lot of proton gradient. So you're going to say we have increased proton gradient.
Or can I just say the pH decreased?
No, because they're saying effect on the proton gradient. So you have to say that name.
Okay.
Okay. You said what? The pH will
Decrease, because there's a lot of hydrogens in there.
Yeah. Also true. Now they're saying justify your prediction. The H+ can't move back to the mitochondrial matrix since ATP synthase was the channel allowing it to do that, right? So ATP synthase, which is this one here, was a channel plus an enzyme. Now it's not working anymore, so now you will be having lots of H plus just stuck in the space. To investigate whether red blood cells of animals lose the ability to take in glucose from their environment as they age, scientists collected red blood cells from guinea pigs that range in age from 1 day old to 7 months old. Scientists incubated an equal number of red blood cells in separate culture dishes that contained a 300 nanomolar solution of radioactively labeled glucose. They like the word radioactively labeled, or the two words. It's like having a GPS on the glucose. They can then track where it's going. The amount of the radioactively labeled glucose present was measured. Perfect. So this is going to be our dependent variable because we are actually measuring it. A. Describe a difference between passive transport and active transport. Very easy. Passive transport doesn't need ATP.
But active does.
Yeah. While active transport requires ATP. B. Justify why the scientists used an equal number of red blood cells in each culture dish. This same question has come back in multiple exams. Why would they use an equal number of cells? This is the best way to eliminate the number of cells as a variable that might affect our interpretation of the results. Because if the results come out differently, you might say, oh yeah, because there were not the same number of cells. So to reduce the headache, use the same number of cells.
So
Multiple
for
answers.
More reliability, kind of.
You should word it in their way. To eliminate the number of cells as a variable that observed difference in results may be attributed to. Okay, very good. C. Glucose transporters are required for the facilitated diffusion of glucose into red blood cells. The scientists claim that the expression of the gene encoding these transporters decreases as the guinea pigs age, meaning more age, the expression of the gene becomes less. If the scientists' claim is supported by experimental data, predict the effect of increased age on the amount of radioactively labeled glucose present inside the cells. So we're told that glucose can enter red blood cells with the help of glucose transporters. But those glucose transporters are made less when the guinea pigs are aging. That means for old guinea pigs, we're going to have less glucose transporters. That means we're going to have less glucose entering into the cells. Okay? Good. So let's write that. Okay, so as the predict just means like just say the effect, less glucose in the cells. D, justify your answer. There will be less expression of genes that code for glucose transporters, so less glucose will enter the cell. The common wild oat is native to regions of Europe and Asia, but is invasive species in central California grasslands. In California, the common wild oat has almost completely replaced some species of native bunch grass. Researchers found that aphids, which is a type of a small insect that often carry plant viruses, have a much higher productive rate in grasslands that include the common wild oat than in grasslands without this common wild oat. You know what this means? The common wild oat, which is an invasive species, will make the reproduction rate of the plant virus higher, or at least the production rate of the aphid that carries the plant virus higher. Additionally, the virus carried by the aphids appears to affect only the native bunch grass and not the invasive species. Native bunch grass infected by the virus will have more death rate than the ones that are not infected by the virus. A. Describe the change in the resilience of an ecosystem, which means resistance of the ecosystem or the strength of the ecosystem. When there is a decrease in the number of the species, we're going to have less resilience because less species means less genetic diversity. So we're going to say less species equates to less genetic diversity, so less resilience of ecosystem. B. Explain how the addition of the common wild oat affects the number of the native bunch grass plants that can be supported by the California grassland ecosystem. Well, this is an invasive species. So what it's going to do is that it's going to take the resources, the space, and the food away from them, resulting in less numbers of them. Okay? But they're saying explain. So you must say what I just said, not predict. So we're going to say that the common...
Go extinct if it does that.
Possibly. It might go extinct, but that's like... Extinct is very extreme, you know? I would just say decrease is safer for you. Common wild oat will consume resources and take up space that native bunch grass plants need. So this leads to less number of native bunch grass plants. Researchers suggest adding ladybugs, which are predators of the aphid, to the California grassland. Predict the effect of adding ladybugs on the abundance of the native bunch grass population. Well, think about it this way. If we were going to have predators of the plant virus carrier, which is the aphid, will now decrease in numbers, which means less plant virus, which means more abundance of the native bunch grass population. Correct? So we're going to say the native bunch grass population will increase in abundance. Justify the aphids will be eaten, so less plant virus carried to the native bunch grass population, so less death of this population. Question 5. Researchers study mechanisms that enable or prevent speciation. A. Describe a post-zygotic mechanism that prevents gene flow and therefore enables speciation. When they say post-zygotic, that means post-mating. So the mating already happened, right? And the offspring is born, but the offspring will not survive, right? Maybe it has developmental issues. Maybe it will survive, but it cannot have kids, just like the case of the mule. So we're going to say the hybrid offspring is sterile, which means infertile, so it cannot have an offspring of its own. New genes can evolve from non-coding regions of DNA. It is not until certain regulatory elements are present in the DNA that a non-coding region becomes a new functional gene that encodes a protein. These regulatory elements include a promoter, a 5' UTR, a start codon, which is not shown here, and also a 3' UTR. Then a stop codon, which is also not shown. Okay. What they're saying is only having exons is not going to be enough to have a functional gene. You must also have those other things that include the promoter, the 5' and the 3' UTR. Researchers studied the evolution of the family of anti-freeze glycoprotein gene, AG gene, in Gadidia, which is what? A family of marine fish known as cods. When present in the fish, these glycoproteins reduce the freezing temperature of the fish, meaning that those fish can now survive better in a colder temperature. The researchers compared genomic sequences in 9 cod species and also with 1 non-cod. So this non-cod is going to essentially serve as our control. Now, they recorded the presence or the absence of the elements of functional AG gene as well as AG-like sequences that are similar to the functional gene but have undergone some sort of a mutation and don't contain all the elements required to enable protein production. Okay. All right. So they recorded the presence or the absence of the functional AG gene or AG-like sequences. Okay, so all of these guys here have the AG-like sequences or the AG repeat sequences. Okay. But some have lost the functionality of the gene and some still have the functionality of the gene present. As you can see, the ones with the still present gene are able to survive in a colder temperature. B. Based on data in figure 2, explain how changes to the genome enabled cods to survive and reproduce after a period of freezing temperatures between 10 and 15 million years ago. So now, let's just look at what happened. You see, between 10 and 15 million years ago, there was a period of freezing temperature. Therefore, for the fish to actually survive, they must have a functional AG gene. And like we mentioned earlier, and also looking at the graph, to have functional AG gene, you must have the promoter, you must have the AG repeat sequences, you must also have the 3' UTR. Okay? So we're going to say that the genome acquired promoter, AG, repeat, sequences and 3' UTR that enabled the AG gene to be functional. Okay. To reduce the freezing temperatures. Now C, using the template in the space provided for your response, place an X on the phylogenetic tree to represent the origin of the functional AG gene. The origin. So origin means it could be today lost or it could be today present, right? So that means you're going to be putting an X on the line that leads to these 6 species. Right now, if you actually trace those 6 species, they would meet up where they would all meet up here. So this is where they diverged. That means you must put an X on this place. This X represents the origin of the gene, and then they would diverge. Some would lose it and some would keep it. So an X there would be perfect. D, based on figure 2, explain how genetic differences among the species on the Gadidia family determine the habitats in which they can survive. Well, the ones with the functional AG gene are able to survive in colder temperatures, and the ones that have lost it cannot survive in those colder temperatures. So we're going to say that the species with functional AG gene can survive in freezing temperatures. We're going to say 1.2 to 6 Celsius. While those without the gene cannot. Scientists can quantify, which means they can measure the rate of translation as ribosomes move along an mRNA from one codon to the next. Using a procedure called ribosome profiling, the scientists measured how long a ribosome remains stationary at each codon of each mRNA. They determined the average translation rate across all codons is 5.2 amino acids per second, but the average translation rate for certain codons in different mRNA sequences can vary widely. Okay. So, these variations in translation rates are thought to facilitate, which means to help correct folding of the protein being produced. The rate at which 3 different codons were translated was measured in 100 different mRNAs. So the rate at which different codons were translated, that's going to be our dependent variable because they are measuring it. The scientists determined the distribution of rate, number of times each rate was recorded for each of the 3 codons, JAK, AUU, and UGG. Okay. A. Using the data in figure 1, graph A to be specific, identify the rate in millisecond per codon that was recorded the greatest number of times for the JAK. Which rate was recorded the most number of times? Which means which one is the highest peak? This is the highest peak right here, which is the 150. So we're going to say 150. millisecond per codon. B. Using the data in figure 1, graphs B and C, describe the variation in translation rate of the AUU codon compared with the UGG codon. So, which one has more variation in the rate? Which one has more spread out data?
The bottom of the UGG.
Exactly, they're so spread out compared to mostly focused in one area. So we're going to say that there is more variation in the UGG translation rate than in the AUU translation rate. Okay. C. Scientists hypothesize that tRNA molecules that bind to UGG codons are available in lower abundance than are tRNAs that bind to AUU codons. Support their hypothesis. So, tRNA molecules bind to UGG codons in less amount. Support their hypothesis. Well, let's just look at the following information. If we have less tRNA, we're going to have less translation rate. Let's support this hypothesis by comparing UGG to AUU. So UGG to AUU. Yes, it does make sense because if you look at the translation rate for the UGG, it's going to be lower than that on average than the AUU. I mean, look at how high those peaks are getting for the AUU. Okay, so basically we're going to say that the translation rate of UGG codon is less on average compared to that of AUU codon. D. Amino acids can be encoded by multiple codons. In many organisms, certain codons for the same amino acid occur more frequently in the mRNA than do other codons. Yes, it's true. If you look at the genetic code table, some amino acids have like 4 codons, 3 codons, maybe even 2 codons, but some codons are simply occurring more frequently than other codons. Explain why the use of one codon over another for the same amino acid might result in increased level of protein production. Well, I think we can tell here that certain codons are simply translated at a higher rate than other codons, correct? And so if they're translated higher than other codons, of course, they would appear more, okay? And so we're going to say that certain codons are translated at a higher rate than others. So this leads to more protein produced from such codons.